worse ways to meet than this.
Hi, well, thank you all very much again. We're now going to start right at 1:30, so we're plugged into Linda's crew in North Carolina and elsewhere, people who are in listening to this one way or another. The procedure for the afternoon, Linda is going to chair the session from 1:30 to 3:30. And at 3:30, we had agreed to sort of play it a little bit by ear and see what would be of people's pleasure. And a number of comments were, well, why don't we begin a little bit of a discussion about where we might go from here? What are the some of the policy questions that are raised by all of this? And so at 3:30, for about a half an hour, 45 minutes, we thought we'd go, Boyd and Gray and I will sort of chair that session and ask from all of you, what do you think are the policy issues that emerge from this? What should we be thinking about on the way to our next session on the 20th of May and then further on down the road? So be thinking about that as this session will be over at 3:30, then we'll spend a half an hour, 45 minutes beginning to block out a little bit of what we might do in May. So again, thank you very much for coming, and to our host and chair, Linda, thank you.
Well, thank you, Tim. Really happy to see so many of you back after that very nice lunch. We already got a little bit of a taste and a little introduction into the potential health effects that we might get from ultrafine particles as well, excuse me, as well as fine particles this morning. But the focus of our panel this afternoon is going to be what are we learning about the effects of ultrafines and associated PAHs on human health in and different concentrations. I think what we're hoping to hear from our presenters this afternoon is more again about what the health effects may be and how they may be a potential predictor for policymakers. I hope that we'll be able to make this all accessible to those of you in the audience or on the web who are not scientists. So we've asked our panelists to be very careful there. So our first speaker is going to be Dr. Ricky Pereira of Columbia University, who is a professor in the Mailman School of Public Health there. She's internationally recognized for pioneering the field of molecular epidemiology, utilizing biomarkers to understand links between environmental exposures and disease. And I should mention that Ricky was among the first, so the association between PAHs and cancer. And now as director of the Columbia Children's Environmental Health Center that NIEHS co-funds along with the EPA, Ricky has done groundbreaking work in looking not only at PAH exposure on an individual basis in women and their children, and looking at a variety of different health outcomes associated with the PAH exposure that these children largely have from some indoor air, but also traffic pollution. So, Ricky.
Thank you, Linda, and thanks to the organizers for putting this meeting together. It's been very, very interesting. I've learned a lot. So this afternoon, I will talk to you about the effects that we have been observing in our cohort of mothers and children in New York City, the effects of prenatal exposure. Sometimes we look at cumulative prenatal and early postnatal exposure to polycyclic aromatic hydrocarbons. And I'll focus on neurodevelopmental impacts, but I'll show you that we're also looking at other adverse outcomes in our studies and have seen links between PAH and several of these, as well as with neurodevelopmental outcomes. And we were among the first national centers established jointly by NIEHS and EPA in 1998, and we've been going ever since. So our oldest children are 15, followed from before they were born. Okay, now this is just this thing, right? Yeah. So the background to the context of my talk is the growing burden of neurodevelopmental disorders in children. The fact that 15% of children in the U.S., 3 to 17, have at least one serious neurodevelopmental disorder. ADHD, learning problems, and intellectual disability, and that the global prevalence of child and adolescent mental disorders is 20%. rates of these conditions have increased both in the US and in Europe and in China. And we know these are multifactorial diseases and not ascribable to any one particular factor. Genetics is important, so are social stressors due to poverty and trauma. But we now understand that environment is playing a role, and our job is to really understand that role and in conjunction with the other factors. A point that I think is sometimes not made strongly enough is that there are certain populations that have disproportionately high exposure and bear disproportionately high risk, disparities in health outcomes and also in levels of exposure. And that's true for PAH. There's a trend to citing more sources of PAH in lower income communities of color. It's sort of the NIMBY phenomenon. And those sources are, for example, let me talk about New York City, where we are doing our studies. This northern Manhattan and the South Bronx are areas that are heavily interlaced by roadways, heavily trafficked. A lot of trucks and buses travel those routes. Most of the diesel bus depots and sewage waste transfer stations in New York City are placed in northern Manhattan, and that leads to a disproportionate exposure. A point that also is not often made is that outdoor pollutants do penetrate quite readily into the indoor environment. We don't just close doors and windows and nothing gets in. So there is a ready exchange. So lower income communities not only experience more exposure as a general rule, but also more material hardship due to poverty and psychosocial stress. And we need to understand their interactions to be able to intervene effectively. Sources of airborne PAH, as we call them, are you've seen these pictures before today, and you know them well. So buses, trucks, cars, automobiles, both gasoline and diesel, power plants and other industrial sources, and indoor sources, secondhand smoke, and that's nice, and and that's okay, and cooking using fuel indoors. Not so much in our country, but grilling of food. So when we do our studies to look at airborne PAH, we need to take account of these other sources, dietary and from smoking. PAH are extraordinarily versatile if you look at the list of the effects that they can exert, both neurodevelopmental, asthma is an outcome associated with PAH in our studies and others, immunotoxicity, carcinogenicity, and obesity. PAH are both genotoxic and known as endocrine disrupting chemicals. The genotoxic aspect is that they form DNA adducts, and at the top of the screen you see a picture of an adduct. This is benzoate-pyrene in this cartoon, lodged very persistently and in the groove of DNA, and that can be measured down to low levels of exposure using a highly sensitive fluorescent or immunoassays antibody techniques. But they're also what's called endocrine disruptors. They can mimic or block steroid hormones, and that's very important because when you're thinking about early brain development, you think of the role of estrogen in that process, where estrogenic pathways are involved in apoptosis, region-specific apoptosis, or causing certain cells to die. They're involved in connecting nerve cells in synaptogenesis, and even in the structure and function of those nerve cells. And so PAH, like benzoate-pyrene, affect gene expression in the hormonal regulatory pathways, and that's been shown in a number of studies, and including in the particular areas of the brain, the hippocampus and the cerebral cortex, that control learning and memory. Now, getting to our study, it's in New York City. We enrolled over 700 mother-child pairs during the mother's pregnancy. And we did not include smoking mothers because we wanted to rule out that very high exposure to PAH and to other chemicals. There are 2,000 plus chemicals in cigarette smoke. So we didn't want to be swamped by that well-known cause of adverse birth outcomes and neurodevelopmental problems. they're African American and Dominican because those are the populations, predominant populations in the areas that we serve in northern Manhattan and the South Bronx, and these red dots are the residences of the women in the study. And here you see one of the newborns in the study and being brought in for examination at 1 year, and here's a girl at 14 also coming in for a visit. Recruited during pregnancy, maternal urine and blood collected, and prenatal personal air monitoring is done with a small backpack monitor. I'll show you a picture. And then we collect biospecimens, cord blood, placental tissue, and then blood and urine as the children are getting older. And we are hoping to be able to follow the children through adolescence. That's a big goal that we have. We take repeat measures from pregnancy on repeatedly over the years of early childhood and into adolescence. We not only do monitoring, personal monitoring and home, indoor, outdoor monitoring, but we have questionnaires that are administered repeatedly. And here I've just put in black the biomarkers that relate to PAH, the DNA adducts, I showed you the picture earlier, and the metabolites that can be measured in urine, a suite of the different metabolic products when PAH are inhaled and processed. But note that we are also studying pesticides and these other exposures, and then looking at some of the changes that occur, immune changes, chromosomal changes, DNA methylation, et cetera. I'm not going to go through everything, we'd be here for weeks. And I will also focus only on neurobehavioral development and not talk about the other outcomes that we're also looking at. There's widespread exposure in our cohort. If you look at the monitoring data, the mother wears this backpack with an intake kind of tube here in the breathing zone, and that's worn or carried with her or near her for several days over pregnancy, and it's been correlated with longer-term monitoring in the indoor environment. The 100% had measurable exposure to PAH in their air samplers, and the range was over several orders of magnitude, wide variability depending on where the woman lived and certain of her behaviors, how much time she spent in various activities. 40% of the cord and maternal white blood cells had detectable levels of adducts. All the mothers and virtually all the children have detectable metabolites in their urine, and usually a number of metabolites, not just one. We've looked at behavioral problems, cognitive problems in the cohort using a number of methods. We've used the Bailey scales for the very young children, ages 1 to 3, and then the various Wechsler intelligence tests, the preschool intelligence, and the WISC for older children, and then a screen for behavioral problems, the child behavior checklist, and then the Connors test for ADHD, and then many, many more, which I haven't listed. The first finding that we reported a number of years ago was an association that was significant between prenatal PAH levels from the backpack monitoring and developmental delays in the cohort at age 3. A child's being examined here in the picture. High prenatal exposure was associated with lower developmental mental development at age 3. And among the most exposed children, the odds of having a developmental delay were about 3 times the odds of children who had less exposure. And as we do in all of our analyses, we do adjust for other factors that could confound our relationship and cause us to draw a conclusion that the causal factor was PAH when in fact it was related to these other factors that are correlated with PAH and with the outcome. So you can see them listed at the bottom. And they include the quality of the home caretaking environment, exposure to secondhand smoke, and a pesticide that we've seen to be a neurotoxicant, and so forth. I probably have to speed this up. How am I doing? Well, you'll
Sorry,
give me.
I forgot to time it. You were so good.
You'll give me the hook.
I think you're okay.
Okay. Great. All right. So then we looked when the children were 5. We looked at their IQ using this Wechsler preschool intelligence test, and found that high prenatal levels of PAH were associated with lower full-scale IQ and lower verbal IQ scores, and they were significant for both of those. In the darker blue, This is high PAH exposure. In the darker blue, you can see that the IQ score is lower than in the less exposed children. So higher PAH levels, lower. Same pattern for the verbal IQ and the same for performance IQ, but not statistically significant. Again, adjusting for these different covariates that could confound our relationship. And the decrement in full-scale IQ is on the order of what's been reported for low-level lead. And of course, we consider that to be an exposure that requires intervention. So we're in the same range in the lower bound. Okay. Then when the children are 6 to 8, we administer the child behavior checklist, and higher scores for the syndrome scales and for the, pardon me, the clinically oriented, what's called DSM scales, indicate more problems. So you can see here that we use 3 different measures of PAH exposure: the air monitored exposure, the maternal adducts measured in mother's blood at delivery, and the level of the same adducts measured in cord blood. And if you look at this particular syndrome scale of anxiety and depression, you see that all 3 are associated significantly. A p-value of less than 0.05 is considered statistically significant. And for the more clinically oriented problems, the same relationship, but most significant for the PAH, for whom we had higher numbers at the time of the publication here of this report. And again, adjusting for covariates as is important. So, and there were also some similar effects on attentional problems in the children. So an indication that these early prenatal exposures could be linked to behavioral problems in the children a number of years later. And then I had asked earlier, I'd mentioned the point really about the possible interactions between pollutants and psychosocial factors due to disadvantage, such as poverty. And we've asked, does the combination of the two magnify the effects of each exposure? And the reason we do that is that understanding that kind of interaction will help us to design more effective interventions to protect the most vulnerable, and then everybody will benefit. So this question we posed was, does material hardship, which is a measure of the family's inability to meet their basic needs for housing, clothing, food, does that exacerbate the effect of PAH exposure occurring prenatally, and here as measured by the adducts in cord blood as an individual biomarker or dosimeter of exposure prenatally. And the child IQ was assessed using the Wechsler Intelligence Scale for Children. And here we see that most of the effect is actually seen among the children who experienced high material hardship. Now, this is a relatively small sample size. I didn't put the sample, but it's less than 300. And so really these are kind of exploratory interactions, but they're important because they show this pattern that it's among the high material hardship that you see the greatest effect of high PAH exposure, with a much higher IQ score, better score, for the low adduct, low exposed group, and a much lower one for the high adduct group, and same pattern for the working memory. So that is suggesting to us that these interactions could be important. And we have investigations of mechanisms ongoing using several approaches. MRI, the children have been, a pilot study has been carried out already, and then a much larger study is ongoing with all of the children being invited to participate in this MRI study at ages 9 to 12. And there's an interest in using MRI to do brain imaging and understand the neural substrate of potential effects of PAH. And I've just shown you, this has no relationship to PAH, it's just a picture of an MRI results from another study. And then another approach we're using is to do epigenetic analyses. I'm just about done, which we are doing, and then parallel laboratory studies. And we have a mixture of PAH that was made up by Lovelace Lab for us to mimic as closely as possible the mixture that is breathed in New York City. And the advantage there is we have access to target tissue. This work is policy relevant. We got a very nice letter from the mayor, Michael Bloomberg, ex-mayor, saying that our findings inspired numerous initiatives that benefit New Yorkers and encourage their ongoing commitment to reducing traffic and other airborne contaminants throughout the 5 boroughs. And our prenatal monitoring has shown a decline from 1998 to 2006. And a very quick glance at a study in China where we've looked at the same pollutants, PAH, using adducts as the dosimeter, adducts in cord blood. And in this study, in this case, it was a power plant. And in this first cohort, the plant was operating. The second cohort, it had been closed. And so the idea there was to see whether we could see a difference between the 2 cohorts. And indeed, the adduct level in the first cohort, I'll tell the good news, second cohort was significantly lower than in the first. So were air levels of the different PAH. The mean level of the neurotrophin BDNF in the children's cord blood was significantly higher in the second cohort, and that's a good protein to have. It's a neurotrophic factor that is very important in early fetal brain development. And we no longer saw associations between exposure and lower developmental scores, which we had seen in the 2005 cohort. So to wrap up, we have evidence of continuing effects of prenatal exposure all the way through age 9, implications for children's academic performance, lifetime earnings, ability to contribute to society, some evidence of interactions with psychosocial stressors, and evidence of direct benefits of policy change. And I must acknowledge my many colleagues in this work and collaborators and our funders, NIHS and EPA, and the Families and Children.
So our next presentation will be by Dr. Beata Risk, who is actually chair of the Department of Epidemiology at the UCLA School of Public Health, and also is appointed in Environmental Health Sciences and Neurology at UCLA. She is actually co-director of the NIH-funded UCLA Center for Environmental Studies of Parkinson's Disease, but Beata is also looking at the other end of the spectrum, looking at neurodevelopmental disorders as well. And Beata.
Yeah, so I actually look at both ends of the life spectrum, but today I will only present the early life. And the research I started in 1995, almost 20 years ago, was on adverse birth outcomes in the LA area, linking them to air pollution in LA. And why did I do that? Well, I found this one study. I mean, looking around LA, it's obvious you should do air pollution studies. And it was also obvious that there was already a large study on asthma funded at the sister university, University of Southern California, so I definitely didn't want to do asthma. And I found this old paper from 1997, published by a UCLA anthropologist who looked at different areas, and this is how really crude it was. They split up LA into these areas, pockets of air pollution, graded these pockets, and assessed all births in one UCLA hospital of mothers who came from these different areas, and all the mothers had to be non-smokers. And they found that the ones who lived in the more polluted areas, which I think were generally towards the east at the time, their children were on average 300 grams lighter than the children of the other mothers. 300 grams is almost 10% of the birth weight, and it is as much as if you were smoking 2 packs of cigarettes throughout pregnancy. That's what we know now, women who smoke about 2 packs. That was the air in LA, in the bad parts. Interestingly, in 1995, nobody had followed up on that, and this is a publication from 1977. And when I wrote my first grant and proposed to look at birth outcomes, the reviewers ridiculed it, saying Dr. Ritz still hasn't learned that babies don't breathe the air while they're in the womb. So, yes, they didn't breathe air while they were in the womb. So why would we think that actually fetal development, and we just heard from Dr. Pereira, it is affected? Why would it be affected? Well, I was a junior professor, had no funding for this, really no funding, but I had databases. And so I linked the birth certificates to the air monitoring stations that were very generously funded by the California Air Resource Board. And you see this kind of Swiss cheese pattern here of the air monitoring stations. I drew a circle around them and assigned pollution during pregnancy. And this was individual for every mother. I figured out when the pregnancy happened, when the first, second, and third trimester were, and what the pollution was that the mother, if she lived at this residency, was exposed to. And so I presumed the fetus through the placenta. And so you would plug in here a mother's home, and then you can draw these circles around it. And what you see here is that when you had, and the marker I had were only criteria pollutants because PM 2.5 wasn't even measured at the time. So as a marker, I chose carbon monoxide. And I chose carbon monoxide because there's a little bit of biologic sense to that because of carboxyhemoglobin, although I didn't believe it was high enough ambiently to really harm the fetus. But I started using CO, and you can see here that above 5.5 ppm average during that trimester, you see a 22%. So for all of you who don't know these numbers, just 1 point, get rid of the 1, and that's the percentage it gives you of increase, okay? 22% increase risk in term low birth weight if you were exposed at this level of CO. Later, when we were able to actually not just look at zip codes of mothers, but at actual addresses, I was able to go down to a 1 mile radius around stations, and you can see now it's a 36% risk increase. So the closer you are to the station, the less exposure misclassification, the larger the value. The less noise, the better your measurement. So we have up to 36% increase in low birth weight risk due to carbon monoxide. Oops. And that was term low birth weight, and you would say, well, term low birth weight, is that really relevant? Well, we have also a California birth defect registry monitoring system, and they were gracious enough to let me use their data. And here we have first, second, and third months carbon monoxide exposure, and these are children who have a hole in their heart, ventricular septum defects, cardiac heart defects at birth. And you can see that specifically in the second months of pregnancy, you see 60%, 97%, 2.8-fold risk increases in these kind of cardiovascular defects if the mothers lived at addresses with these high carbon monoxide levels during the second months. Why is that important? Because we don't see it in the first and third months, because the heart is actually formed in the second month. So there's a lot of biologic specificity in these measurements. And I'm talking about CO. Aren't we here to talk about particles? Well, in the meantime, the Particle Center was funded, and my colleagues were actually moving their measurement instruments away from the 405 freeway. And you can see here, this is downwind. And these are the measures for particle number, black carbon, and carbon monoxide, and they almost fall into the same line. So what I had done by choosing my criteria pollutant was I was honing in on ultrafine particles and black carbon. So CO in LA really represents proximity to sources. The RIOPA study was going on at the same time, and this is what we measured. We asked them to put a CO monitor into the homes, and you can see these are correlations of indoor and outdoor CO with distance from the freeway. And you can see that indoor and outdoor at certain times of the day where there's rush hour, they again correlate very nicely if the home is closer to the freeway. This would be on the freeway, and this is up to 300 meters from the freeway. The same results that we saw with these downwind measurements, just moving outdoor equipment away. This is inside homes. So the same story, 300 meters, you would think that's what, 750 feet. That's not a lot of distance, right? We can breathe well if we are beyond that. Well, one of our colleagues recently, actually, these were all daytime measures, measured at night. And at night, these kind of pollution indicators actually went out to 1 mile. So in 1 mile in LA, you're definitely near a freeway somewhere. But you're not only near freeway, you're actually also near lots of very highly trafficked roadways. So the black is the number of cars on our freeways. You see the black lines is really the freeways because they're the most cars. But you see other roadways that are pretty dark purple and blue. They also have a lot of traffic, such as 50,000 to 70,000, 75,000 and more cars per day. So we know in LA you drive, and in LA you have traffic and congestion. So it's really a picture where we know it's all over and all around us. So the next step we did was to actually plug in the address of the mother and draw a circle around it and then see how much traffic there was around the home. And this was in the mid-1990s, where you could still use that traffic measure. And this is for all births. You see an increase with the number of quintiles of traffic density, but it's especially high for preterm births here in the winter births. And why is that? Because the air gets trapped under the inversion layer. And locally, you have much higher exposure where you are when it's trapped where you breathe, which is at road level. And these are some of the measures that we then published for preterm births. Again, about 30% increase in risk for preterm births in LA. And this is also a measure that we adjusted. And now all this stuff I showed you before was not funded. But then NIEHS gave us money. We went out and interviewed 2,600 women and asked them all the questions that my colleagues had always raised, which was, but isn't it parity, age, race, education, smoking, alcohol use, anything you can think of, indoor air pollution? Well, we went and asked women, we measured it, we threw everything in our model, and the effect did not go away. If anything, it strengthened. So it was not confounding that caused these exposure measures to show that there's an effect. This is just a meta-analysis that I did with a young colleague that showed that overall there is probably a 15% at least increase in preterm births due to PM 2.5 measures in this case. And I wasn't satisfied with the traffic density because we knew that it was really the composition on the roadway of cars that should be mattering, as well as industry, et cetera, or road geometry. And so with Dr. Mike Jarrett, we set out to build this kind of land use regression model. These are all the monitors we hung in order to validate the model, 200 all over the basin. And you can see that our model predicts, as you would imagine, the roadways quite nicely. I'd shown you that roadway with traffic map before. This is an air pollution map now from our model, and you can see the road structure in LA quite nicely. So we're using this this land use regression model, as well as a dispersion model that was made public by, called the CalLine 4, the California Line Source Dispersion Model, that actually uses also meteorology and emissions data. But everybody kind of has a problem with the emissions data being appropriate. So we have now 2 additional measures. We not only have traffic density, we have our land use regression model that we built, and this emissions-based model as well. And when we use all of these, you see that they confirm what I'd seen before. So this is for preeclampsia, which is a serious condition during pregnancy for women that actually the mother has to give birth preterm in order to survive a lot of times. It's a very serious condition. And with the CalLine 4 emissions model here for NOx and particles of 2.5 size, you can see that the odds ratio is really increased up to 40% for this kind of condition. Preterm births, you see similarly, we can now compare. Finally, so the 2000s arrived, PM 2.5 was finally measured by EPA. We now have measures for PM 2.5, and you can see here that yes, we get a little bit of a signal for PM 2.5 for preterm births, But these confidence intervals include 1, so they are not significant, maybe a 4%, maybe a 9% increase. But then when you use our land use regression models for traffic, you get the 5% with a tighter confidence interval. You get the PM 2.5 prediction from the CalLine emissions model, 4% risk increase per interquartile range. Traffic density, maybe 2%, maybe nothing. There's not much there. We also learned that these models work well in LA, but not in Orange County. You have to rebuild them. You can't just transport them from one place to another. That's another story. Luckily, California Air Resource Board also measures air toxics, and this was our chance in those 2-year periods where the MATES study worked in the mid-2000s. We're actually able to look at PM composition. So this is organic carbon, elemental carbon, nitrate, ammonium nitrate, PM, biomass burning, and diesel. And you can see here that they are all increasing risk, 13 to 21% or 12 to 21%, all statistically highly significant. We have almost 7,000 cases and almost 40,000 controls. So these are large numbers. And these effects are all there, and they're there for no matter what kind of composition you're using here for preterm birth. We then went back and analyzed data that a colleague of ours had collected in the mid-1990s on size of heads. And this is a fetal head, and this is the biparietal diameter that we use to measure head size. And you can see here when you're to the left of this line, this is that it's smaller. And you can see our CalLine model predicts a smaller head in the exposed. Our land use regression model predicts it. Ambient CO predicts it, and ambient NO2. But we believe that these are all wonderful traffic markers. So the head size was significantly reduced at birth, which goes quite along with what Dr. Pereira has just told us in this cohort. And this was also a minority cohort, mainly minority, so vulnerable population as well. That said, we have now also data, because everybody in California is concerned about autism. We have now data from the Department of Developmental Services, and this is the largest study done in California, where we link the birth certificates to the LA DDS database. So we had about 8,600 cases. So this is not a small study, and 80,000 controls. And you can see when we're using our LUR land use regression model for traffic, you see a 9% increase in risk. And a PM 2.5 monitored, you see an 8%, both very significant increase in risk of autism. So an almost 10% risk increase for autism. You would say, does that really matter? Well, if you think about 8,000 children, that would give you 800 children times $3 million that every autistic child costs you. You get quickly to $3 billion. So this is not cheap. Again, all of this was adjusted for lots of variables. So this is, this is my summary slide for these results, where you can see that probably fine and ultrafine particles, but definitely traffic-related exposures predicted term low birth weight, cardiac heart defects, head size, and autism. And we believe that roadways and traffic was a main contributing source. And this is just reminding you that Beijing is certainly worse off. And they had in 2013 this not great news to report of infants being smaller. And what we did after that is say, well, we have now these amazing exposure models for LA and for California. Why not apply them to other diseases? And this is one other disease we just applied it to, again with NIEHS funding, in this case for a pilot study. It will probably be the most published pilot study ever. It already has 8 publications in the last 3 months. And what we can show is here that when you do the exposure assessment right, for example, use our LUR model, use the CalLine emissions-based model, you can see that for entire pregnancy exposures, you see 9% increases in acute lymphocytic leukemia. For the CalLine model, it predicts a little less, But then for the eye tumors, retinoblastomas, that are also related to the brain tissue, you see 14 to 19% risk increases. And for germ cell tumors, those are tumors that are from developmental origins, 16% risk increase. When we then use our air toxics, let's just, so that was for PM 2.5. With the PM 2.5 monitoring, you can see similar kind of increases, but the confidence intervals were wide. We have a lot more variation in exposure. PM 2.5 is just a mixture. We never really know what we get, whether we really measure the toxic agent or not. So what we did is go to the air toxics monitoring stations again, and they are all over California, about 28 of them. And here you can see for total PAHs, and this is now retinoblastoma and leukemia. The first one is ALL, so that's leukemia. For all pregnancies, you can see we have 17% risk increase with PAHs, 7% for benzathipirine, and another pyrene here, 12%. And then benzopyrrolin, 60% risk increase. For retinoblastoma, again, the eye tumor, almost 70% increase. And for Wilms tumor, a 15% risk increase. You could also see we were able to not just look at all pregnancy, but third trimester, and it's consistent. And actually, the larger effects for the leukemias were all in the third trimester, which kind of makes biologic sense again. And all of this would not have been done without the generous help of lots of people who worked with me over the last 20 years, and the California Air Resource Board money to build the LUR model, and of course NIEHS. That's it.
Thank you very much. That was a little concerning. So our third speaker this afternoon is Jim Zhang, who recently moved near the southern part of heaven from the University of Southern California. He's now at Duke, where he had been at USC. He'd been a professor of environmental and global health and the director of the Environmental Biomarkers Analysis Lab. Jim's work actually, he was very involved in some of the climate change work, where he was actually officially recognized in the 2007 Nobel Peace Prize awarded to the IPCC. And he's currently focusing on looking at some of the biological mechanisms by which environmental exposures exert adverse health effects. Jim?
Thank you, Linda, and it's a pleasure to share some of my ultrafine particle research findings here today. Let's see how this goes. Let me start with this study that was done a few years ago, funded by Health Effects Institute. And this is a study that we're looking into real-world exposure to urban traffic mixture. In London, this is the natural exposure chamber in Oxford Street, central London. And it's a very busy commercial tourist place. During the daytimes, the private-owned cars are not allowed to travel. So it's only the diesel, the buses are powered by diesel, and also the official taxi cab, that black official London cab you can see. They are all powered by diesel engines. So sort of pretty a good diesel and rich environment in this street. So we designed a study to use that in comparison to a nearby park, which are relatively clean. You can see this part of the park. You don't see busy roads inside of the park. So we have pretty much a very straightforward study design, just like we have a controlled exposure chamber, but in this case it's naturally in two places. And we invited study participants to walk for 2 hours in each places. And this is one day that study staff had a few subjects walking in Hyde Park. So it's a 2-hour exposure that we measure lung function. Those are asthmatics. They have either mild or moderate asthma by clinical definition. So we measure their lung function at other biomarkers I will show you later before they start a walk and in the middle of the walk, which is at a 1-hour interval, and then hours after the walk, up to 24 hours when we invite them to come back to the clinic to do the measurements. So what we find is that the lung function, we use FEV1 and FVC, those two parameters of lung function drops by 6% after 2-hour walk in the Oxford Street compared to the separate day they walked in the park. And we tried, so we clearly seeing a effects on the lung function, and we try to understand why their lung function get reduced after that 2-hour walk in a street with lots of pollution, with lots of particles. So we measured the inflammatory biomarkers in the airways. Those are the things I show here, proteins, some lipid peroxidation products, and cells that are reflecting pro-inflammation in the airways. So we do see that those changes are associated with the lung function reduction. So that indicating that those people's lung and get increased inflammation at oxidative stress after they walk in the street. And then, of course, we all see this as a mixture. You have even noise, you have particles, you have gases, that sort of things. But we do measure ultrafine particles. We measure elemental carbon. We measure NO2, PM 2.5, and we will try to see which of those pollutants are more strongly associated with the lung function reduction with the various biomarkers we measured. And it turns out that UFP was associated with the more markers than the others. Now, then the question is whether UFP is really responsible for those adverse effects, or the UFP is a better marker for the mixture that those subjects were exposed. That this study itself, I think we cannot really answer that question, but we do put out those associations. There's need other studies to really answer those questions. So then let me move to next study, which will try to answer those questions, because this is sort of lecture that when I teach air pollution health effects to undergraduate or graduate students, this is where we started to tell people where, when particles are inhaled, the really sizes would determine how far they can travel and how fast they can be cleared inside of the airways. So this is a study, in the study that I'm going to show you in the next few minutes, is try to understand how particles will affect the heart and also affect the lung. So we focused on, for the lung, we're looking into the inflammation, we're looking into the airway oxidative stress burdens, and in the heart, we'll look into the autonomic function, the heart rate, blood pressure, that sort of thing. And also, we're looking into cardiovascular effects, looking into coagulation markers, and also looking into systemic inflammation and oxidative stress. So sort of looking into various biomarkers that are related to those four pathways, which probably underlying pathways for cardiovascular and pulmonary diseases, and to see what sizes is going, which of the particle sizes will be related more to each of those pathways. So I'm not going to go into the details, but this is the 4 pathways I showed you. There is those molecular markers we measure, try to see how things change. So this study was done also in a real-world setting during the Beijing Olympics. This morning, I'm glad that Stacy have already showed you about the air pollution changes during that period, because the control measures were very, very aggressive and tried to bring down the air pollution levels in Beijing when the games were in town. And then everything went back to normal after everybody left the town. So that's we designed this study, try to measure people. This is panel study. We recruited the healthy young people, so with the hope that the other factors will not complicate observations. So in this case, now we measure them 2 times before, 2 times during, and 2 times after the Olympics, which really relate to those control actions. We started this in a relatively short time period from beginning June, end of June, mid-June to end of October. So all the things that will not really will not change too much if we look into intra-person comparison, because we measure same people just at different time points. And this is just to show you that indeed, this is a 3 pollutant measurements, PM 2.5. You can see there is a reduction that medium, that line come down for about 35% during the Olympics. That's the red line. And then the ultrafine particles come down a little bit more than 35%. If you look at the median, that's about 50%. Benzoyl A-pyrene come down actually a little bit similar to the ultrafine particles. But there is a reduction there. There is a huge variability you can see because of the natural day-to-day variability. So this paper published in JAMA showing the cardiovascular-related biomarkers, that this paper published in the American Journal of Respiratory Critical Care Medicine, showing the pulmonary biomarkers. So the details you can find there, but pretty much so this is not all the biomarkers we have measured, but just to look at the biomarkers, majority of the things, you have those changes. Many of those markers, when the levels coming down, that means the inflammation levels get reduced, coagulation tendency got reduced, so it's a good thing when they come down. Some going up a little bit better, like airway acidity. Anyway, lots of those biomarkers, we do see a change, significant, and then that indicating that those cardiopulmonary pathways are actually getting better when the air quality is better. And the other way, when post the Olympics, many of those biomarkers got reversed. So that's the message. Now the next question, especially related to today's talk, is to look at the role of UFP, ultrafine particles, and PM 2.5. So first of all, if we look at ultrafine particle, this is a number concentration, of course. At PM 2.5 is the mass concentration, which definitely driven by accumulation mode particles or little larger particles, because that's mass measurements. You can see the correlation is UFP at PM 2.5 is not that large, and it's negative. And so that's a good thing from an epidemiological standpoint, because if you find association with the UFP with a biomarker, and you also find UFP with the PM 2.5, they're not necessarily from, indicating the same thing, because they probably will be independent, because a UFP at PM 2.5 will not go correlated. So this is a complicated slides, but showing different biomarkers, that the changes of the biomarkers per unit change of the pollutant. You can see those bars, if they are away from the vertical line, that means they're statistically significant. So the take-home message is that PM 2.5 UFP both show changes, significant changes with many of those biomarkers, but because two of them are not highly correlated, they are actually very poorly correlated and also negatively correlated, so their effects could be independent. And then, as I said, we understand that from a physiological standpoint, that UFPs are cleared more slowly because they can be trapped in the deep lung longer than the larger particles. So we would expect the effects in the lung probably will last a little bit longer. This is where we really find the red lines. Those are the lag days. 0 means the day after the exposure. 1, 2, 3, 4, 5, 6 days. That's each days after the exposure. So that's called lag. So you look at that lag pattern. A UFP with this biomarker, this is a pulmonary inflammation marker, and you can see that UFP association sustained for a few days. PM2.5, like a 0 lag, coming down quickly, so indicating probably PM2.5, which mass is really dominated by accumulation mode particles, probably cleared faster than the fine, ultrafine particles. And then the other thing is that we think that some of the systemic effects were spillover of lung effects. So if the ultrafines are stuck in the lung longer, so the systemic effects probably were also more prolonged. And then if you look at that, actually, for this marker, this is 8-hydroxydeoxyguanosine, which is a stable product for DNA oxidation by reactive oxygen species. You can see that ultrafine particles did not really get an association until 3 days. So maybe it takes some time to explain the spillover of lung effects to a systemic effect, which is a hypothesized mechanism for PM effects. Then the last thing we observed is that we always say, well, ultrafine particles, so they're so small, so they almost act like a gas. So they probably can directly shoot into the circulatory system, not like particles will deposit in the lung, and then larger ones deposit in the lung, and then have the cascading effects. But ultrafine particles just probably like, when they're very small, they probably can directly go into the bloodstream and cause responses. So we do have one biomarker here. This is a white blood cells, and some epi studies did show that air pollution exposure is related to white blood cells. And in this study, we can see that ultrafine particles, we do see association as day 0, day 1. But if you look at PM2.5, we really don't see that. So sort of support that PM ultrafine particle deposited, cleared differently or translocated differently from larger ones in the body. So this was just this paper was just accepted last week. So details is in that paper, and the study was funded by NIEHS, Health Effects Institute, and Chinese Natural Science Foundation, and the Beijing government. Without the study subjects, we won't be able to. I won't be able to report these findings. Of course, it's a very intensive 6 weeks or 8 weeks when we need to collect so lots of data in such a short period of time in 2008 when the games were in Beijing. So thank you very much for your attention.
Well, I think we're hearing about a multitude of different kinds of health effects that can be associated with different kinds of traffic-associated pollutants as other air pollutants. So our fourth speaker this afternoon is Doug Brugge, who is at Tufts University School of Medicine. He actually directs the Tufts Community Research Center and is part of leadership at the Community Engagement Core of the Clinical and Translational Science Institute. Much of his research has focused with community work and has largely employed a model of community collaborative research and I think he's going to talk about some of the associations of asthma and traffic-related pollution.
Thank you. It won't be asthma. Our focus is on cardiovascular disease. Thank you, though, for the introduction. I'm going to talk about the Community Assessment of Freeway Exposure and Health project, which is a series of studies. I'm going to focus on the NIEHS-funded work, which was our original grant, but I just point out in passing that we have additional projects funded by NHLBI, HUD, EPA, and more recently the Kresge Foundation. All right, how do I move this forward? There we go. You just have to push it a little harder. All right. Just to summarize the aims of my talk today, we set out about 6 years ago, and actually, Linda, our no-cost extension expired yesterday, so the countdown on being able to report to you has begun. But we set out about 6 years ago to do the following: to measure ultrafine particles as particle number count using condensation particle counters in 3 near-highway areas and matched urban background areas in the Boston area. To build hourly models, land use regression models of PNC for 1 year for each neighborhood. To adjust the ambient PNC levels at the residences of participants. We set out to recruit participants in these study areas for time activity and other factors that would change their exposure. We now call this time activity adjusted PNC, or TAA PNC, to test associations of time activity adjusted PNC with biomarkers. Our primary biomarkers are blood markers of inflammation, and that's what I'll talk about today, CRP, IL-6, and tumor necrosis factor alpha receptor 2. We also measure fibrinogen, blood pressure, ankle brachial index, and self-reported adverse cardiovascular outcomes. And to do all this within the context of a community-based participatory research study with deep community engagement and involvement. So the starting point for where we began is that we were aware at the beginning that there were many studies that had looked at near highway exposures, and this is just, whoops, I don't want to do that. Okay, in the upper graph, if you're looking at it on your left-hand side, shows the curves for a variety of pollutants near highways or major roadways, and shows that regularly environmental science researchers have found that there is a gradient of these pollutants from the edge of the highway to a couple of hundred to 300 or 400 meters away from the highway, and among those pollutants are ultrafine particles. And that Carner's article, I think, summarized 41 of these studies that had been done at the point where they published their summary. On the other side, you see just one example of a study, a proximity study. This is Hoffman out of Germany, in this case looking at the proximity of residences to major roadways and coronary arteriosclerosis in those participants in the study, and they see increasing prevalence of this adverse effect with more proximity to the roadways, much as Dr. Ritz was presenting about the LA, but I just chose this one because it's really easy to present next to the other one, and it makes sort of a clear comparison. What we wanted to do, because our primary hypothesis was that ultrafine particles are a good candidate as being a causal agent here, we wanted to both measure the ultrafine particles and measure biomarkers in participants and test associations between the two in the same study. So what we did is we chose 3 near highway communities, one in Somerville, one in Dorchester, South Boston, and one in Chinatown. We recruited people, all the green dots are people that we recruited. We recruited people near the highway, and then also people in what we call an urban background area, which is greater than 1 kilometer from the highway. And the urban background areas were matched with each of the near highway areas. We recruited about 700 people total, and then got blood samples from 450, at least one blood sample from 450 of them. Along the way, I had an idea that we should do a proximity study just to see how that looked in our data. We had a lull in data collection and weren't able to analyze, data wasn't ready to be analyzed. I thought, let's do an extra paper, but that ended up being a lot of work. So I've been telling my, you're nodding your head, thank you. I tell people on my team to slap me the next time I say something like that. But anyway, we did publish this late last year, and in fact, we did find associations with proximity, although not the neat higher near the highway and then declining with distance. It's a lot flatter and actually a sort of an odd lower level of elevation of CRP and of IL-6 in these two graphs in the 50 to 150 meter group. I won't go too deeply into this, but just to put one little point here, one of the things that seems to be a lesson out of our study is we went in thinking the highway is everything, it's so big, and I think the local traffic and the major roadways are also significant, and that's one of the things that's going on here. In addition to recruiting participants in these near highway communities, we also purchased an old recreational vehicle, tore out all the stuff inside of it and replaced it with air monitoring equipment. This slide shows the equipment that we installed in the vehicle. The thing that we're most interested in here today is the PNC, the condensation particle counter, the TSI CPC, as is listed here. But we have measured a bunch of these other pollutants. We have not had the resources to analyze the other pollutants significantly, so I will focus on the PNC. And then on the far side there, you see that we have a GPS, and the goal here is to be able to link the measurement of the pollution with geographic location. And in fact, with the PNC and the GPS each having 1 second measurement times, we get very, very large data sets over our study areas. And this is just to show some of that raw data. This is from 1 day in our Somerville study area, and you can see each of the panels is a different hour moving forward in the day, and the large black line is the highway, and then the sort of satellite area farther from there is the urban background area. You can see that, and then red color, the darker the red, the higher the PNC levels, and you can see that PNC is generally higher near the highway and declines with distance away from the highway, and is also higher early in the morning, which is what we usually see, especially on a colder day, early in the morning, and then declines over the course of the day. So this is just to show you that we did succeed in collecting this data. We went out 40 or 50 times a year in each neighborhood. So we have data sets of millions of data points of PNC for each neighborhood. 1 year, and then we went out at different times of the day and different days of the week and through all of the seasons. So we have very large data sets of the PNC data, but also of the other data, but the analysis of the PNC. And then we had a great graduate student who just defended her thesis, Allison Patton, who used this data to build regression models, predictive models of PNC. And let me just say here that building a predictive model of PNC is problematic and is difficult, and maybe there are reasons no one did it before we tried to do it because it's so difficult, and maybe you just have to not know how crazy that is to go and try and do it. But anyway, she has succeeded. She has models, and I'll show you in a moment, for each of the study areas. But the reason this is so difficult is that the PNC levels are changing rapidly in time and space. A difference of 50 meters can be a substantial change in PNC. A difference of 7 a.m. versus 10 a.m. could be a significant difference. And then, and we'll get to this secondarily, not only are the PNC levels changing rapidly, but the people that we're trying to study are moving. That we tried to get them to stay still and to not go anywhere for a whole year, and none of them would agree. So they're moving. They leave home, they go to work, they go shopping, they do all kinds of crazy things, and And so they are not in a fixed location, and so assigning exposure is a substantial challenge. And let me just point out the contrast with PM 2.5, which is a regional pollutant that is sort of generally spread over a larger metropolitan area, where you can do studies where you have a central monitor and you could draw a circle of a couple or 10 or 15 kilometers around it and assume that people largely got that exposure and probably stayed within that study area most of that time. So that's the central challenge of our study. So Allison went and actually did build these models. This is just 2 slides from her paper that just came out in Environmental Science and Technology that illustrates what she was able to do. And the solid lines, let's see, on your left-hand side is a winter morning. On the right-hand side is a summer morning. So we always see higher PNC levels in the winter than we do in the summer in colder weather. The solid lines are the actual measured values of the RV driving around that fixed route on both of those days at those hours. And then the triangles are the locations of our participants with the value that is predicted by her predictive model assigned for that hour of that day of that year, all right? So I want to make sure you're clear about this. What she is doing is predicting an hourly value for every residence of our participants for every hour of the year. So 8,760 hours in a year, that many values get fed out of her model and then delivered to the next step, which is the next graduate student who then works on them to assign exposure, all right? And what you can see from this is, the models are not perfect. In fact, very few people have tried to do this before us. There are a couple of other regression models of ultrafines in the literature. Our R-squareds are about the same or in similar range to theirs. They're not as high as you can get with the NO2 models by any means. But, you're getting values that are in the right direction here and are roughly comparable. So, okay. And then this is the figure that took 6 years to produce. And what I like to say to Allison is it brought tears to my eyes when she first showed it to me because I knew how much effort was behind this. But this is, and I want to just point out for anyone who's media or anything, this is not published, so this is off the record, and it could change before it's published. But what this shows is the ultrafine levels in the 3 study areas, and I know it looks like 4 because one of the urban background areas is far away from one of the study areas, Chinatown and Malden is the urban background area for Chinatown. But you can see that in Somerville and in Chinatown, the near highway areas have much higher levels. This is the annual average. You take that 8,760 hours in the year and average them. This is the value for every area within the study areas. Malden, the urban background, is lower. And then the Dorchester study area, we for a long time had a question of what is wrong with Dorchester in our team meetings. And what is wrong with Dorchester, it appears, is that we did not see the same kind of gradient in Dorchester that we saw in the other areas. In fact, near highway was not as high as we had anticipated it being, which is a little bit of a problem for us. But also an interesting observation that we chose 3 near highway areas in the same metropolitan region along the same highway, and Somerville and Dorchester, if you look at them, look almost exactly the same, and yet one of them is profoundly different in terms of its exposure metrics, which is interesting too. I think I'll skip that one for this. And then Allison passes those along to our next excellent graduate student, Kevin Lane, and he modifies these exposures based on the individual characteristics of each participant. And what is up here is, again, what I said, that the UFP concentrations vary greatly over space and time, and people move around. And this graph is showing time activity, and what the red is time indoors at home, the light green is outdoors at home, the blue, the darker blue is time at work or school, and then the light blue is other, and then the line is time spent on highways. And this is just for the people who are employed or full-time students. We have a separate graph for the people who are unemployed. and are not in school. And what you can see is for the people who work, there's a big chunk of the day when they're not at home. And what that means is that we should not be assigning them the residential exposure during that time period. And so what Kevin does is for every hour of the day, based on their time activity patterns, he assigns different values, substitutes basically the urban background for the time when they're away from home, and gives them a different exposure. And this is, this is very interesting because most air pollution research assigns the ambient, the outdoor annual average level at the home as the exposure. So we are not doing that. We are dividing it up by hours of the year and then assigning them different exposure. We also have values for when they're on the highway, for example. And he also modifies this. If they're indoors, they have central air conditioning and the air conditioning is on, which drives the levels way, way down indoors. We know that from another paper one of our students did. This is just a map that shows the assigned the time activity adjusted exposures for all the participants in the study. And so you can see that has been done. This next slide really shows the effect, though. What's up there right now is the Somerville study area. It's the annual average ambient assigned at the home, no adjustment done. And you can see people living near the highway with the yellows and reds have higher exposures, and the urban background with the light blue and dark blue have lower exposures. And when he does the time activity adjustment, we get a differential exposure misclassification. That is, the urban background doesn't change very much, but many people near the highway have lower exposures. And the reason that is exactly what I was saying before. They went away to work, okay? Some of them have a light blue. There are a couple of points there that turn to light blue. They are gone during the peak times when exposure would be happening. So if you just assign them the annual ambient value, you would assign them the wrong exposure, and worse yet, not just would you be assigning the wrong exposure, you would be differentially misclassifying the exposure of the people nearer the highway relative to the people in the urban background. And that, in an epidemiological sense, is a serious problem. That should say in preparation. Some of this has been published. And then what Kevin is doing is building structural equation models, and I'll just throw this up there to show you what we're doing. Building constructs, traffic air pollution, tobacco smoke, socioeconomic status. The yellow boxes are the actually measured values. The PNC, it should be time activity adjusted PNC annual average, job exposure. Then down below for tobacco smoke, secondhand smoke, direct smoking. SES is composed of income and education, and then systemic inflammation from the blood inflammatory markers. And this is just with the Somerville data. This is still a work in progress, but shows a statistically significant association between the PNC and traffic air pollution, and then with traffic air pollution and systemic inflammation. These are also controlled. The model's adjusted for age, gender, and BMI. So that's the direction we're going with this. I will just mention very briefly, we have a problem that we're just starting to address. I mentioned that Chinatown was one of our communities, and the Chinese immigrant population is, it turns out, this is a paper that's under review right now. The Chinese immigrant population is lower education, they're immigrants, they're non-English speaking, they're low income, all those things, and yet most of their health markers are vastly superior to the rest of the study population. And so we're trying to figure out how to deal with that right now. Maybe am I out of time? Just about. Can I say one last thing? Okay. Let me just mention the Kresge funded work because I think people here would be interested in it. This is not a research project. They funded us to look at policy and practice. And this is a list that one of our community leaders, Wig Zamar, put together for us. But these are tactics for reducing near highway pollution exposure. And we are looking at tactics for building construction, for siting buildings, for siting playgrounds and pathways. We're looking at possible policies that we could put in place in the city of Somerville. The city government is a strong collaborator with us and is very interested in doing this. And so we are exploring, we have a charrette scheduled for about 5 or 6 weeks from now, where we will bring designers and planners and engineers and all sorts of people together to look at 2 near highway sites that are proposed for development and to think about how they could be done in a more protective way. So we're moving into the realm of policy and planning implementation with this foundation funding. Okay. And then just also to mention, if you want to know more about us, because we're a community collaboration, we have a really great website. If you put CAFEH, I was saying to somebody at lunch, it's my only success with acronyms and names ever in my entire life, but it's a fabulous success. If you put CAFEH into Google, it's the first site that comes up. We have all our publications there. We have lay fact sheets. If you're not into reading technical science, we have lay fact sheets, not just in English, but in multiple languages, and all kinds of stuff there that you could go and look at, all right? And because we're a collaboration, there's been over 100 people who've worked on this project, and I just throw that up to make sure no one thinks that I did all of this or something, okay? Thank you.